Thank you, Anna, for the kind introduction. I lost you. And thank you all for joining us in this session. I'm going to tell you about our research into a certain type of cancer. As you know, cancer is really an umbrella of complex diseases, and all of them have different mechanisms, usually in how they work and how they develop resistance to drugs. So we are focusing on multiple myeloma, a type of blood cancer that affects the bone marrow. It's a disease that causes a series of symptoms. Basically, it thickens the blood, also causes susceptibility to infections, which is the main cause of death, and also causes susceptibility to bone lesions. So treatment of multiple myeloma has undergone many developments over the years. The current standard treatments started around the year 2000, and there's a series of treatments that are applied as standard to any patients. There's a bit of variation depending on some characterizations that we can make, but there is definitely a certain standard. And we are particularly interested in the development of resistant mechanisms to the newer treatments that are available, starting in about 2015, which includes immunotherapy and such. So myeloma patients usually undergo this cycle of responding, hopefully, to a certain treatment, but they will undergo relapse for a while, and again, resistance and relapse, and this is kind of the cycle that we are seeing. Okay, so myeloma is not curable right now; it's treatable, but really response depends a lot on the patients. This is what we're trying to understand. Also, there's a huge variation in clonality in the disease because the same treatments are favoring other clones developing, and then you're changing again your target. And this is kind of what we are trying to really understand. Characterize this clonality, if you want, and try to deal with it. So there's a lot of work that was done based on single nucleotide variations, and because they were more available, because the technology was there, because our knowledge was there, but we firmly believed that epigenetics and larger aberrations, structural aberrations, would open the key to much more insight into treating these diseases. So to us, R10, the newer chemistry / flow cell, was kind of the promised land, because I personally was not interested in targeted approaches. I wanted to try to tackle the problem in a whole genome kind of point of view, right? So we really needed to have the capacity to sequence a sample in a single flow cell for it to be kind of cost effective, with the idea of scaling it up to clinical trials eventually. That was kind of the dream. So R10 made this possible, at least in principle. We wanted to test it by ourselves. So to be safe, what we did was start this analysis with cell lines, right? We wanted to have ideal conditions for nanopore sequencing, so we wanted to have unlimited material, quote unquote. So what we had is 3 cell lines that they are in some or other way resistant to different treatments, which is not necessarily the most important part, except for the last one. So I'm going to describe them. The first one is the ERP1 cell line, which is intrinsically resistant to some of the classical treatments. Then there is an induced knockout of BCMA, which is a gene involved also in the type of resistance that was induced by CRISPR. And finally, that knockout was made resistant to one of the classical then treatments, bortezomib, by simply exposing it to gradually increasing doses of the drug. So we started sequencing these cell lines with ideal conditions, and we saw that indeed we could reproduce what was promised. So we were getting 99% accuracy and lengths that were what we were expecting based on the fragmentation that we'd been before. We were hoping for 10K, we were near to 9K, so it was working fairly well for us in these conditions. Now, the key question was, can we do the coverage, right? We wanted to go for something around 30x to be sure that we could call everything we wanted. We wanted to go for the whole omics coverage. So we wanted the big variations, we have the small variations, and the methylation. So 30x was like the sweet spot, and we were able to get pretty close. In some cases, we actually exceeded it, some cases falling shorter. But we were able to actually get to this at least baseline of 20x that we were hoping for. So now we had data, we wanted to see if we could pick up signal, and we took
what I call an exceedingly naive approach to try to see it, and we based this on modifications, methylation, right? So what we did is we did the base calling and the methylation calling with the standard tools, and then we took CPG annotations from the UCSC browser, and we did differential methylation between our different cell lines, see if we could pick something up. And we defined our interesting cases as the ones that the two first cell lines, so ARP1 and the knockout, would keep the same methylation level, and then there would be a variation on the bortezomib-resistant one. And we were hoping that this would kind of yield some results to confirm that we did have some signal in data. And this was happily the case, and within the first couple of hits, we found a downstream effector of CD42, which has been documented as being related not only to myeloma resistance, but specifically to bortezomib itself. Right? So this gave us a lot of hope as the first approach to looking at this data. Seemed to kind of say that myeloma, methylation signal was strong enough to pick up interesting cases. First check. In order to go into detail, we want to confirm that this differential methylation was actually present in the data to double check. So we create this kind of plots. I want to drive your attention to the bottom signal, which is a smooth signal of the methylation, where you can see that the light blue line representing the bortezomib resistant has hypermethylation compared to the rest of the samples. And this is what is causing that differential annotation that we found. The thin lines that are seeing there, I'm not sure you can see in the screen, there's thin lines in that region on the right, which represent regulatory regions, okay? So promoters and enhancers, which is exactly what we are after seeing and characterizing. So methylation seemed to give a signal. Now we were wondering about structure variation, which was actually my initial motivation before switching to the very strong methylation signal. And we wanted to see some basics. So this is a complex plot. I will try to drive your attention to the important bits, which is basically the little black squares in the middle. Those are the CRISPR sites where we did the knockouts for the knockout and for the resistant cell line. So you cannot see them in the first one, completely white, but in the other two, you actually can pick up the knockout nicely, nice confirmation. But then interestingly, there's a full gene inversion that's happening on the bortezomib resistant one. Now, this could be just a clonality change, so we're favoring the ones that where the CRISPR caused this inversion and it's really associated to this enrichment, or it could be something else, so we're looking into it. There's some indications that it could be something else and a bit somehow interesting. So the cell lines worked to establish that we could see the things that we wanted to see if we had enough material, but the next challenge was going to clinical cases and seeing if we could also do interesting things with these kind of more challenging samples. So we got our hands into 3 clinical cases that underwent different steps of treatment in myeloma. All of these guys, girls, whatever they are, cases, underwent the standard treatments for myeloma. Okay, so we are actually seeing them go into resistant into the new immunotherapies. Okay, so CAR-Ts, bispecifics, and these are the ones that we want to tackle and see if we were able to actually look at them. So a bit of an anecdote here. This eventually turned into the Lord of the Rings projects because I couldn't pronounce elranatamab, and Lara was calling it Elrond, and that drove one thing led to the other, and this now is the Lord of the Rings projects. So for the nerds in the audience, maybe you find that at all interesting. Anyway, coming back to the science. So we were able to sequence these samples. These were more challenging. We not always were able to reach 1 microgram. Usually we couldn't. So half of this or even less. And in all of them, we achieved eventually 20x coverage. In some of them, I think it was 2 or 3, we actually have to use some Nanopore black magic and suck up the sample and load it in a new flow cell. I think this was only 2 cases, so still very reasonable. And if 10% of what Clive describes comes true, this will not be needed in the near future, so that's exciting. So another check we want to do is, can we reproduce previous results from our own group? And yes, so Lara characterized the cerebron gene as being involved in myeloma resistance, and we can pick up this signal in our own data. So it shows that we can reconstruct the treatment history of these patients by looking at these methylation signatures, for example. So where are we now? So we are trying to find good candidates, interesting candidates from
this methylation data. And we have picked up a few genes already that are showing differential methylation patterns that could explain, could suggest a mechanism of resistance, and we are actively looking into it. So far, everything we've thrown at them in terms of the classical ways of looking at things like facts seem to correlate with the signal we're seeing. So this looks very, very promising. This is ongoing work, and hopefully we'll have more to say about this very, very soon. So with that, I'd like to acknowledge our collaborators, and thank you very much for your attention.